Hello and welcome, and thank you for joining me today. Today I'm going to be talking a little bit and updating mostly about where we're at with the future of biocompatibility testing with animal-free or in vitro testing. We're going to spend a lot of time talking about the in vitro irritation. That's the one that's the most exciting right now and on the verge of global acceptance. But we're also going to be talking about some other in vitro alternatives that are just right there on the cusp of being accepted, or the ones that we've already been using a little while now to get rid of animal testing. Obviously, none of us love to do animal testing. It is needed in certain circumstances. But today, we have science that's progressed so far that we're able to really try to get a good idea of the safety of these medical devices without having to use animals. And so we were just trying to convince our regulatory agencies around the world that these tests can adequately represent the risks of medical devices to a human by using in vitro alternatives. So for the standard today, we're going to be talking and using most of these two kind of standards and guidance documents. So for those of you that may not be aware or is familiar with biocomp, first off, we hope you fall in love with biocompatibility through this presentation. But these are the two main kind of documents that we use for biocompatibility. We have the ISO 10993 suite. So this is the ISO standard. It consists of multiple different sections that all cover different endpoints for biocompatibility. -1 is kind of like an overall guidance on how to do a risk-based approach for biocompatibility. And then each different section has to do with a different endpoint, like sensitization or irritation or cytotoxicity. And recently, we actually split out. So 10993-10 used to be irritation and sensitization, and they were combined together in one standard. Because we now are moving towards having an in vitro irritation, we actually separated that standard out. So -10 is going to stay sensitization. And so it's only going to include, once we redo it, sensitization information. Now we have a 10993-23, and that standard is going to be just for irritation. And in that standard, we added the first kind of thought process for irritation should be in vitro. And we'll cover what that looks like here during this presentation and where we currently stand. But then we also have the FDA's guidance document. So in Europe, the ISO suites, the ISO standards are kind of like a law. So as a notified body, they'll look at that standard and they'll interpret it, but they'll basically follow it as they evaluate the device acceptance for a CE mark, and they'll just follow it kind of word for word. So if the standard in Europe for the -23, if it says you should consider in vitro irritation first, your notified body is going to want you to consider in vitro irritation first before you jump to an animal. The FDA doesn't necessarily have to follow 10993. In fact, a lot of times they only recognize portions of the standard. So what they do to try to help is they release a guidance document, and that guidance document is their interpretation of those standards, how they interpret it or how they're going to enforce it. And so the good part about this is you get a little bit more insight on how the FDA actually thinks about the standard. The downside of it is it takes a long time for the FDA to kind of give out this guidance documents. So there's things that can change, like an acceptance of an in vitro irritation test, for example, that they won't get to updating the guidance in time. So the guidance is just that, it's guidance, it's not a law. Most of the time they'll try to follow their guidance, but there can be some differences how the FDA is moving compared to what the guidance says. But those are the two standards we're going to follow today in that presentation. So when we start looking at some of these previous and why this is becoming so important to consider in vitro alternatives, this slide represents the old kind of approach to biocompatibility. So this chart on the right side of the slide, this chart was from the G95. So this was the original FDA guidance document that was created in 1995, a long time ago. And the older I get, I realize that is a long time ago. But in this guidance document is pretty easy. This is how biocomp was thought of a long time ago. And honestly, even today, we're trying to change this mentality. But what you do is you look at this chart and you look at the device contact the body. You look at how long it's in contact with the body. So where is it contacting? How long is it contacting? And then you go through it and you just look at what tests are required. And this is how we did biocomp. It was really easy, actually, and we didn't need a whole lot of biocompatibility experts because anybody who would ever learn how to follow a chart could do biocomp. It was just sometimes you had to kind of maybe have a misinterpretation or agreement or disagreement on how the device contacted the body or the duration. But once you agreed on that, everybody knew what test to do. So this was what we called a checkbox mentality. So you just basically did the tests regardless of any other considerations. You would, fill out a test request form to your favorite CRO. You get the test results back. As long as everything passed, you submit it to the FDA or your notified body, and that was your biocomp. So easy peasy. And that was how we did biocomp for a very long time. The problem with this type of approach is that you didn't have to understand the materials of construction on your device, and you didn't have to understand anything about the testing itself. So these devices, like as long as you passed these tests, you could put anything in these medical devices. You didn't even have to know anything about them, right? So we had people who would go to local hardware stores and buy screws and things off the shelves and put it into an implant because it passed these tests. And as far as not knowing anything about the tests, most of these tests have been around since the 50s and 60s. So if we thought 1995 was a long time ago, think how the world has changed since the 50s and 60s, but yet these tests have not, right? So we're using these really antiquated tests that don't really relate very well to how the device is going to be used or how the human body is going to react. And so it's really an antiquated kind of approach that we're trying to change very quickly in the ISO 10093, and that includes in vitro alternatives. So the new standards are getting away from this checkbox approach, and they're going to what we call a risk-based approach. And I know that this is kind of not directly related to in vitro, but I'm going to tie it here in just a second. So this is the new chart, the one that replaced that old one that we showed you. And as you can see, we have the first category or the first column we added here was physical and or chemical information. So the concept here is before you do anything with your device, you need to understand the type of information or what is going in it materials wise. So what kind of physical information or chemical information of your materials and processing of your device? That's required. So you have to do some kind of research or understanding, testing, or something to help you understand the physical or chemical information. And then there's some things we changed. I'm not going to go through this. There's other seminars or webinars that we've given to go through this in detail. You can look up. But the important part, the reason why I show this, why it ties to in vitro, is because we also took away all those X's. So as you can see, the only one that has X's are that physical and chemical information. Other than that, the other thing we changed everything to E's. So X's are prerequisites, so you need that information to do a risk assessment, determine what testing you need. The E, now look at that paragraph we put in for E, and we did this on, if you ever on a standard committee, you know how long and how much discussion goes into just one paragraph, right? So an E means endpoints to be evaluated with a risk assessment through existing data, additional endpoint specific testing, or rationale. And so we put that in there because we didn't want you to go straight to the animal tests. We want you to think about, does this test even need to be done? Can I do other ways to evaluate this, right? So can I do this through a risk assessment? Is my device just titanium with a cleaning validation? You know how many times titanium has been tested and we know it's pretty safe. So do I need to go through all these animal tests if I need to, or my device is safe just based off of the processing and material it's made out of? So if you look at it, if a device is manufactured from novel materials, not previously used in medical device applications, and no toxicological data exists in literature, these additional endpoints could be done by testing. But if you do know a lot about these materials, if you do have really good process understanding and cleaning validations, then we have a low risk of that medical device. And so why do we want to jump straight into animal testing? That should be the first thing you look for to try to reduce animal testing. And it just happens to be reduced costs and turnaround time too, because you're not doing tests. In this case, you're doing assessments. But I think so many people are still checkboxing that they jump right into tests. But no, these are endpoints to be evaluated, and there's lots of ways to evaluate those endpoints, including in vitro alternatives to those animal tests if you don't feel like you have sufficient information to justify out of them, okay? So that's the first way. The second way that we're currently getting rid of animal testing is actually using extractables and leachable data. So here we have these endpoints that I talked about before. And if you look at these red arrows, these are the endpoints that we could utilize extractable and leachable data, if it's performed correctly, with a toxicological risk assessment to justify the risk of those the biological endpoints. So to put it probably more bluntly, if you do extractable leachables with a tox risk assessment, you don't have to do all these categories that the arrows are pointing to. The majority of these categories, except for genotoxicity, are animal tests, right? So we can get away from doing animal testing by doing good extractable leachable testing and toxicological risk assessments. So again, even if you do an initial risk assessment and determine that you do not have enough information to just justify these endpoints, these endpoints can be addressed using in vitro alternatives that are accepted globally as an alternative to the animal tests. And when I mean globally, we've submitted to US FDA, all the notified bodies, even Japan has started to accept these, China, so Canadian FDA. So we've done a pretty good job getting global acceptance of extractable leachables. So keep that in mind, is that we're limiting how much animal studies, even if you have to do all these endpoints, even if you can't justify out of any of them, we've already knocked off a lot of these by doing E&L and tox risk assessments. And the reason we can do this is because when we look at these systemic tox endpoints that we mentioned where the arrows are pointing, all these tox endpoints have to do with something called a dose-response curve. So what you want to look for is most of these chemicals have what we call like an S curve. Now, there are some that has a little bit of an upswing at the very end of the tail, or the beginning of the tail. So for example, this is caffeine. And we all know people who maybe haven't had their caffeine in the morning, and there can be some irritability. So the beginning of the dose of caffeine, if you don't have any caffeine, if there's zero, there can be some adverse response. So this is a lot of drugs. For example, like if someone needs a daily dose of a drug, if you're not getting that drug, there's an adverse response. And then when you get that drug, like caffeine in the morning for most of us, then you get to a new normal. Then you can become pleasant and you can actually talk to your spouse or significant other or partner or just friends and not have a mean conversation. So that's where things are called the new normal or just your baseline dose response. But then once you start getting higher response of this drug or this compound or this chemical per body weight, you start to get some other types of bad responses. Like you start getting arrhythmia when you have too much caffeine. You can actually start having tremors or hallucinogens. And then we get to something called an LD50, which is 50% chance of death. So you're killing half the population at that dose. That's not something we want to get close to, right? And then we have where everybody would be dosed and killed off that compound, and that's the death dose. So every single compound, every single chemical has this type of curve. And so what we try to do with E&L, instead of injecting this into an animal and seeing what kind of response it has, does it have tremors? Does it start to have arrhythmia? All this testing's already been done. It's in the literature. People have done this for almost all the compounds we see off medical devices. So why repeat the studies? Why don't we just see the chemistry that comes off, the amount that comes off, and then compare it to this dose response curve? Where on this curve are we at as far as that compound? That helps us determine if it's a systemic toxicity risk or not. So this is how we go through the E&L evaluation. We have a lot more information on toxicology. If you fall in love with this type of thing, I recommend going and learning more about tox. It's awesome. You can do this dose response curve on sugars, smoked meat. There's lots of great and fun things to do with toxicology. Okay, so the other reason why we love E&L in chemistry and in vitro alternatives is because a lot of times we get better quantity and quality of information from these reports. The turnaround time's not dictated by biology. We don't have to let an animal kind of metabolize or digest these compounds. And so we can do it much quicker usually. And usually they're less expensive. So they're better for the animal, obviously. That we get better data most of the time. They're quicker and most of the time cheaper. And we're starting to get more regulatory bodies actually requesting, for example, E&L because the data is better. So we're trying to take this momentum and turn it into more of this in vitro alternatives to the animal tests we do already that cannot be looked at through E&L. So the main thing that we're working on right now, and we've been working on this for a few years now, but we're just on the verge of getting accepted. In fact, check back soon. I'm hoping that we'll be able to have an update from the FDA. We'll get to that in a minute. But what we are looking to do is one of those endpoints that the arrows was not pointing to in that slide was irritation. And that's because irritation, even though technically there is some irritation data on certain chemicals, they're not always applicable to the type of contact your device has, and it tends to be kind of sparse. So most of the time we have to do irritation testing. Most of the time right now that's done in rabbits. And so it's injected into a rabbit to look for redness and swelling, a typical irritation response. We now have these reconstructed human epidermis skins, which you see down on the bottom right there. These skins are actually grown up in a lab, so they're taken from a stem cell, and they're grown into tissues. And this tissue is actually very good representation of our human skins. In fact, if anything, they're a little bit more loosely tied together. So if you look at the very top of that, the RH tissue skin, you start to see that there's a lot more area or loosely, there's not that dark purple kind of gross looking stuff on top. That allows more of the chemical to leach through the skin and interact with the cells below. And so this actually makes these tissues a little bit more sensitive than our own human tissue, but it's a very good representation. In fact, if you're ever at Nelson Labs or at Salt Lake and you want to come see the lab, you can come and see these tissues. They actually have little fingerprints on them and everything. So they're pretty cool little tissues. So up in the left corner, that's that tissue itself. So we get those in, we order them in. We apply the extract of your medical device to that tissue, and then we look for irritation. And how we look for irritation is we expose them to a salt that's a staining salt. So what happens if it's a viable, healthy cell, they will metabolize that salt and turn purple. So after you metabolize the salt, there's a purple byproduct from that metabolization. So in theory, the healthier and more valuable those cells are, the more purple we'll see in the extract. So all we have to do is expose these tissues to the salt and let them turn purple, or get them to metabolize all that salt. And then we can use alcohol to extract all that purple out of the cells. And then we put it on a spectrophotometer that measures the color that's there. And we can see how much purple is present in each well. The more purple there, the more viable, the more metabolism that happens, the more viable those cells are. So as you can see, those two wells on the left of the plate, they look a little bit purple, and the one that looks clear, the one on the top is our positive control. Obviously, that's going to be one that positively kills the cells. So there's no purple there. The one just below it has a little bit of purple there. That's one of our known irritants. So you can actually see some irritation response happening. So this is a way for us to screen these tissues to see if there's been an impact to the viability of the cells, and this is particularly sensitive to irritants. So when we see this impact, we know that there's been irritation there. There's another step if we need to confirm irritation. It looks for something called interleukins, but this is a good screening step for us to just screen to see if irritation has been possible. Then this is the actual response we get from the spectrophotometer. So as you can see, 50%, that red line on the right, that's our acceptance criteria. So anything above 50% is considered a non-irritant. Anything below 50% is a possible irritant, okay? So then you would want to investigate further, like maybe then you start doing some animal testing to see if you see a response, or maybe you look at what's happening in your system, like is there detergents there that are not being washed away, or some other chemicals known to be irritants that could be left behind. So this is a good screening test for irritation. Now, current regulatory acceptance, and obviously this is going to be based on the current date we are talking right now, because we're hopefully changing this soon. But as you can see, we have a lot of regulatory bodies that currently accept this test. In fact, it is the preferred method in ISO 1093-23. So that means all of Europe is accepting this as its preferred method. We also have China and Korea, or China and Japan accepting it. And we also have other affiliates around the world, CEN affiliates. So a lot of regulatory acceptance for this test. We run it. We have it validated here at Nelson Labs and our lab in Leuven. So we're running this test, all the time. Now, some key countries have not yet recognized Part 23, and this is the part that we're trying to influence right now. Australia, Canada, Republic of Korea, and most importantly, United States. In fact, Australia, Canada, and Korea are probably, and we talked to them, are just kind of waiting for the United States because they're concerned that the United States hasn't accepted it yet and want to know why. So as soon as we get the United States on board, everybody else would probably be on board afterwards. So, and I know the majority of the people listening to this webinar are probably submitting their samples to the FDA sometime in the future. So for them, or if not right away, so for them, this is a concern. Like if they're not accepting the in vitro, I'm stuck doing the animal. And that's true today. So let me talk about a little bit about what we're doing to try to influence this. So we've been working on this for a very long time. We've had probably for the last 5 years a submission into the FDA called the MDDT. It's a process within the FDA where they have to look at new investigation techniques. And we've had this even before -23 was accepted as a standard. We've gone through the FDA multiple times. Nelson Labs with others in the industry has published dual data. So we used, in fact, it's public, so I can say about it. Arthrex, one of our great clients, did a side-by-side comparison of animal and in vitro data on the same devices. And we published that so that the FDA could have a published record to show that the in vitro is actually more sensitive than the animal test. And there was a couple times where we saw a slightly positive response in the in vitro, never saw an irritation response in vivo. Well, the FDA is now asking for more of this dual data. So we are working with AVIMED, and we have about 4 to 5 different companies who have done dual data in the past. And we're now combining that data together to be able to submit back to the FDA for them to look at. So we're literally, I had a call this morning where we were gathering that data and getting it in one spreadsheet, and we're hoping to send it out to the FDA to review through AVIMED next month. So what that means is that the FDA, as long as all that data looks, right now it looks good, but as long as that data kind of supports that the in vitro irritation is actually more sensitive than the animal test for irritation screening, then we're hoping the FDA would take that, and then that would influence them to recognize the new -23. So we feel like we're on the verge of any time now the FDA accepting this standard. And once the FDA accepts the standard, then we believe that other holdouts would fall in line, and then we'd have worldwide global acceptance of the in vitro test for irritation. So that's where we stand. For those of you that are interested in all the work that's been done on this irritation, here are some of the published documents that we've done over the years, not just Nelson, but there's the industry for helping prove that this in vitro irritation test is more sensitive, so a better screening test for medical devices than the animal test. So now that I've talked about irritation and kind of where we're at, we're not done. It's not like we're going to sit back and go, okay, irritation's done. We're done trying to save animals and do better science. That's just not how we work. So we're currently working on sensitization. For me, this is a big win that I want to have before I'm retired in this industry. So for me, sensitization is the one that I feel like has the biggest negative impact to animals and to pocketbooks and to turnaround time for medical devices. So for those of you that are not familiar with sensitization, just very high level, it's called delayed hypersensitivity, type 4 hypersensitivity. So you have these cells in your body. These cells are memory T cells, and they are constantly on the hunt for sensitizers or antigens. And when they identify something that they think is a potential issue, they will recognize it. So a good example of a sensitizer is poison ivy. It's a very common one that people are familiar with. The first time you contact poison ivy, you don't break out. You don't get the redness and swelling, but your body's cells recognize that poison ivy, and now they're on the hunt. They're looking for that antigen again. So the next time you touch poison ivy, or the third time, then you break out. That's why it's called delayed hypersensitivity. Plus, it's systemic. So you could touch poison ivy with your hand the first time and then step on it the second time, and it would break out on your foot. So it's not localized reaction. The reaction's localized because it's just going to stay within the foot, but it's systemic as far as the cell's response go. So that's what sensitization is. Right now, it's done on guinea pigs. This is a very high level of guinea pigs. So I've talked about this at conferences before. In my 21 years working in biocompatibility here at Nelson Labs, I've tested on about 430-something thousand guinea pigs. And in that time, we've had one positive. So another reason why I'm really passionate about getting this test to an in vitro alternative. So the problem with an in vitro alternative for this, the reason we didn't start with sensitization first is because the sensitization reaction is actually pretty complicated. There's a lot more, we know it really well, so it's not unknown to us, but trying to predict it through an in vitro response takes a little bit more effort than the irritation. So, and the irritation test was already established for other industries like cosmetics and food. So there and drugs. So we had some backbone there, but sensitization has also been established by those industries also. So it's not like we're starting from scratch. So there's multiple approaches to be considered, and maybe we do combinations of these. So there's some in chemical ones, which is like this direct peptide reactivity assay. That's one to look at. Some of the ones that I like are the next two, which is in silico. So this one, think about it this way. So this is, we know what a sensitizer chemical looks like, kind of like a structure. I like to kind of compare this to a police lineup, right? So you put up all those chemicals in a lineup, and you know the expert or the person, the witness, knows what one looks like the bad one. And that's what we do with the chemicals. So you run E&L, you put all your chemicals up, and you look at their structure. And sensitizers have a unique structure. And so we can actually compare this using QSAR, which is in that bullet there. It's a quantified structure analysis report. And so we can actually compare the structure of those compounds to a known sensitizer and get an idea if it's a potential sensitizer or not. With that, we also have to kind of have like an evaluation threshold so that we know that below this, no other sensitizer, like it's not going to be a sensitizer if it's below that amount, right? So the dose of it's going to be so low that it won't cause sensitization. So we need to establish that. And then we also have to make sure we validate that this QSAR can work for medical devices. The other side, which, like I said, might be a combination of these, is in vitro alternatives. And there's a lot of work already done and accepted in these types of things. For example, the GUARD assay, the EpiSense, the Sensitox assays, these are all assays that are currently available that they do use in certain industries to predict sensitization. And we could definitely, and some of these have already been proven to be viable to look for sensitization through animal or through medical device too. So the next step for these is actually go through the same steps for irritation, but because we grease the wheels a little bit with irritation, we're hoping that this is a lot quicker. We know the FDA now kind of understands what they need to know. We know what the FDA needs to know. So we're starting right now in the ISO to start looking at these tests, doing some round robin potentials, get some data, published data together, and be able to actually go to the FDA with these types of answers. Plus, my colleague, Kelly Coleman, who is a champion for these in vitro for Medtronic in vitro alternatives, he's actually looking to publish with help for some of us the actual answers to what the FDA was asking for the in vitro irritation. The concept there is we want everybody to know what we went through to get the in vitro irritation accepted. So we want to publish the questions and the answers so that we want more people to try to come up with alternatives to animal testing, and they'll know what they have to do to get the regulatory acceptance through the FDA. Okay, so I mentioned the sensitization threshold. These are things that are being worked on right now. Here's two publications that are proposing the threshold. Remember, this is where nothing above or below this threshold would be a sensitizer, regardless of what it is. This is important because without some kind of threshold, we don't know how sensitive to make our chemistry to screen for it. So we need something to be able to tell us this is how low you need to screen for, and then anything below this, we don't have to worry about. And so these are just two examples. There's actually been a third one that's been published just recently that was really good too. So there's been a lot of good work put towards this sensitization threshold idea. Okay, so, and then this is just, once again, I kind of talked about this, but this is kind of where we're going with sensitization. We're looking to put this into -10. So it's already there right now. We have this this in vitro alternative already in the tables in -10 and Annex C. But we're trying to do more of a round robin approach like we did irritation, get testing labs to try these out, find out which ones make good in vitro alternatives to the animal test. We have to make sure we can both use them in polar and non-polar extractions because that's what medical device uses. It's not like a drug or a chemical that can be put directly on there. We have to have an extraction. So we have to have a method that works in both. So that's what we're doing right now in the ISO committees. So just to get you an idea too, it's not we're going to stop at irritation and sensitization. We also are looking at hemocompatibility and trying to get away from some of the animal tests that exist in there. So, for example, there's been a lot of work on thrombogenicity, which historically has been done in canines. And it's like the worst test you can possibly imagine because sometimes you're done in 2 canines and sometimes the controls in one won't even match the controls in the other canine. So, trying to come up with a better way to evaluate thrombogenicity. There's things like in vitro blood loops, there's Chandler loops, there's all these different things that different companies like Abbott and Medtronic have come up with. There's a bunch of test methods that we can do that we've helped with to get through the FDA. So right now you can do alternatives for animal testing. The FDA will accept certain types of alternative loop models for the canine. You just have to have a discussion with them beforehand with the protocol to make sure that they're on board. So it does require like a pre-sub to go through the protocol with them to make sure that it works, but that this is something that's accepted. And then, we also have things like pyrogenicity, which we're looking for this monocyte activation test as an alternative, or rabbit, looking how we can replace the rabbit pyrogen test. So we're constantly looking to what animal tests can we replace, what other in vitro are out there. We're working as an industry very closely together to try to come up with the best ways that we can do this. So I want to also talk about this pyrogenicity. As you see the last bullet point there, PETA is actually being involved to help us working through their own validation study for an MDDT submission for pyrogenicity. So it's not just the medical device industry that's looking at this. There's a bunch of people who are all working together to try to eliminate animal testing from medical devices. Now, I knew I probably threw out a lot of acronyms during the presentation, so these are just for your reference in case you didn't have an idea why I said it or put it on the slide, just to have your information. Also, I'm hoping you're seeing this on the Academy, but if not, I want to make you aware that the Academy contains tons of this resources. And when we know about the in vitro acceptance, the Academy will be the first place that we put it out there. So please sign up for the Academy. It's all free. There's all this great content from Nelson Labs and Sterigenics and Nordion that we can help fulfill any questions you have for medical device needs. And for that, thank you for your time. And if you have any questions, please reach out. Thank you.